Hello everybody, today we'll talk about lecture number 6 in the course Molecular Genetics MLGN 301 2006. Today's lecture is called Gene Function number 1. In this lecture we will talk about many essential things in molecular genetics, and we have to understand many definitions. Of course, there is a recommended book for further reading. It's called Molecular Biology, 4th edition, by Robert Weaver. In this chapter, actually, we'll see the function of genes and how it works. So we'll learn there is some objectives to learn, and there are some outcomes that by the end of these sessions and practical students are expected to be able to understand. We have to know the protein structure, the types and of protein structure. We have to know the protein function, synthesis of RNA, and transcription landmarks, and stages of transcription, which is start with initiation and then elongation and termination. We have to know the difference between these three terms. The central dogma of in molecular biology and molecular genetics is that the gene transcribed into RNA, and the RNA are translated into a protein. Of course, as we have known from the different from other lectures that we have taken before, that cells express different subsets of genes in different tissues and under different conditions. And of course, we know also from the few last lectures that there are of course regulatory elements that's called promoters, and these promoters are regulate the transcription of genes. And we also have studied that there are different also some genes have alternative promoters. So genes don't have only one promoter, but some genes have alternative promoter, more than one promoter. And we know that the main function of DNA is storing information. So producing a protein from DNA information involves both transcription and translation. So of course we know that a codon the DNA have a codon that is a 3 base sequence that determines what amino acid is used. Template, and we have the DNA have two different strands. A strand called template strand. This template strand is a complementary DNA strand that is used to generate mRNA, and non-template strand which is not used to in RNA transcription. This, as we see, template strand and non-template strand. The template strand is used for transcription, and non-template strand will not be used for transcription. And of course, after the transcription, the translation occur, and then of course producing amino acids for building the polypeptide chain. Protein structure. Then the protein structure happen. Protein are chain-like polymers of small subunits which called amino acids. DNA has 4 different nucleotides, but protein have 20 different amino acids. And each amino acid, all the amino acids have this same feature. The same main feature. There are 4 main features in the amino acids, which is all the amino acids have an amino group. All amino acids have had a hydroxyl group. All amino acids have a hydrogen atom. All amino acids have a specific side chain. So these 4 characteristics could be found in all amino acids. So this amino acid, let's take a look in 4 different amino acids. This 4 different amino acid is simply like C, M, V, L, and these 4 different amino acids have, as we said, amino group like this one here, like that one here, hydroxyl group like that one here, and of course hydrogen, which is not here in this picture, but of course there is a hydrogen atom in all these different amino acid, and specific side chain which is specific for each amino acid, and actually it gives the amino acid the difference between the variation between different amino acids are in the specific side chain, like here C, MVLAD, whatever. So as we can see here, you can see here the hydrogen atom here in this picture. So there is a site which is R, and the hydroxyl group, and the NH group, amino group. Amino acids are joined together via peptide bonds. Chain of amino acids are called polypeptides. Protein are composed of one or more polypeptides. Polypeptides have polarity, as does DNA. Free amino group are one end in the amino, or they call it N-terminus. So where the amino acid, the NH2, where the amino group is called the N-terminus, and the other side of the chain is called the hydroxyl group. At the other side of the end of the carboxyl is called C-terminus. So each amino acid chain have two ends. The first end is called N-terminus, and the last end is called C-terminus. And when we start to read amino acid, we have to start from the N-terminus to the C-terminus. So when you start to read a gene from the 5' to 3', you can read the protein from the N-terminus to the C-terminus. So here we can see amino acids that are joined together, and of course you can see from the NH2 side to the COH side, like that. So if we were to read MAVLD, that's how we can read this chain of amino acids. Types of protein structure. The linear order of amino acid is called a protein primary structure. Interaction of amino acids, amino and carboxyl groups give rise of secondary structure, and this secondary structure is a result of amino acids and carboxyl hydrogen bonding among near neighbors. Common types of secondary structure. There are two types of secondary structure. The first one is called alpha helix, and the second type is called beta sheet. In alpha helix, secondary structure, polypeptide backbone groups hydrogen bond with each other, like this. We can see that this happen between a hydrogen bond of the polypeptide. Dashed line in the figure indicate hydrogen bonds between nearby amino acids. So the nearby amino acid have a dash, this hydrogen bonds, and this interaction between hydrogen bonds resulted in the secondary structure, and this secondary structure have two types, alpha helix and beta sheet. The beta sheet pattern of two structure also occur when polypeptide backbone groups form a hydrogen bond. In the sheet conformation, extended polypeptide chain are packed side by side. This side-by-side packing creates a sheet appearance. So two polypeptide chain can create this sheet appearance. So these are the two main structures that can be seen in the secondary structure: the alpha helix and the beta sheet. So okay, and number 3 is the tertiary structure. Tertiary structure, total three-dimensional shape of polypeptide is its tertiary structure. A prominent aspect of this structure is interaction of the amino acid as side chain. So the side chain of the amino acid, which we have talked before, which we call it R group, this amino acid side chain interaction with each other can result in this globular structure. This globular form of a polypeptide is roughly a spherical structure. This is the tertiary structure. So this, as we can see, this one polypeptide chain can generate this tertiary structure. Protein domains. This is very important to understand the protein domains and the difference between domains and motif. The compact structure of region of a protein are referred as domain. So the compact structure or region of a protein are referred as domain. Immunoglobulin provide an example of 4 globular domains. There are 4 globular domains. Domains may contain common structure, function, motif, so like zinc finger, hydrophobic pocket. These are called motif. So when the domain have a function, we can call it motif. Quaternary structure is interaction of two or more polypeptides. So this is another level. The fourth level of protein is called quaternary structure, which means that this is interaction of two or more polypeptides. So to review again, primary structure is a single strand of polypeptides are joined together in a single polypeptide chain, and of course this is the primary structure. If it has if it confirm a beta, it can confirm the secondary structure can confirm a beta pleated sheet and or alpha helix. This is alpha helix and this is a beta sheet. This confirmation it can cause to produce the secondary structure. The secondary structure, the interaction between the the R groups in different amino acids can cause the tertiary structure to happen, and the tertiary structure of different polypeptide chains can gather together and cause quaternary structure. So we have primary, secondary, and tertiary, and then quaternary structure. Summary of this lecture: we have proteins are polymers of amino acids linked through peptide bonds. Sequences of amino acids is in a polypeptide. Primary structure gives rise to that molecules. Local shape gives secondary structure. Overall shape is tertiary structure. Interaction with other polypeptides called quaternary structure. Okay, now we talk about the protein function. Protein function: proteins provide the structure that helps give rise of cell integrity and shape, serves as a hormones that carrying signals from one cell to another, bind and carry substances, control the activities of genes, serves as enzyme that catalyze hundreds of chemical reactions. All that are different aspects and different of protein function. Okay, now we'll see the relation between genes and proteins. As we know that in 1902, Dr. Garold suggested a link between human disease and a recessive gene. If a single gene controlled the production of an enzyme, lack of that enzyme could result in the buildup of homogenetic acid, which exerted in the urine. Should the gene responsible for the enzyme be defective, then the enzyme would likely to be defective. One gene, one polypeptide. In this regard, the theory is that over time many experts have built in Garold's initial work that many enzymes contain more than one polypeptide chain. This means that each polypeptide is usually coded by one gene. This means that in this observation have led to the theory of one gene, one polypeptide hypothesis. Most genes contain the information for making one polypeptide. As we see that this means that the protein is information carrier. In 1950 and 1950s and 1960s, the concept that messenger RNA carries information from gene to ribosome developed, and then an intermediate carrier was needed as the DNA is found in the nucleus, while proteins are made in the cytoplasm. So some types of molecule must move information from DNA in the nucleus to the site of protein synthesis in the cytoplasm, and this of course leading the discovery of messenger RNA. And as we know that the ribosomes are a cytoplasmic site of protein synthesis, and the RNA from ribosomes does not move between the nucleus and cytoplasm. Anyway, the Jacob proposed an alternative of non-specialized ribosome that translate unstable RNA that are called messengers. And these messengers are independent RNA that move information from genes to ribosome. So all this historical introduction is that about the discovery of the RNA, which we know that now the RNA in the messenger RNA is the one that take the information from the gene from inside the nucleus and this information is translated into a cytoplasm by the ribosome to produce a protein. And we have known now the four different levels of protein structure. So all that is just from previous slides, we just have to know that the messenger RNA carried the genetic information from genes to ribosome, which then synthesize polypeptide. So now we'll talk about different the main three different steps in transcription. Number one, in transcription, coding sequences can be transcribed to RNA. As we know that the coding sequence is transcribed to RNA. This is transcription. And RNA nucleotides similar to DNA, slightly different, of course, the backbone, except the uracil instead of thymine. Okay, the transcription now follows the same base pairing rules as DNA replication, but remember, U replaces T in the RNA. This base pairing pattern ensures that the RNA transcribed is a faithful copy of the gene. For transcription to occur at a significant rate, its reaction is enzyme mediated. So this means that it needs enzyme to let it happen. So that's why the enzyme directing transcription is called RNA polymerase. So the transcription will not happen without the RNA polymerase to attach to the beginning of the gene. As we can see that how this happened and how this transcribed, this figure can describe how this the building of transcription and how that could occur since of RNA. So now come to the main point of the lecture, the transcriptional phases. So we have three different transcriptional phases. Transcription occurs in three phases. Number one is called initiation, and number two is called elongation, and number three is called termination. In initiation, we can see that the RNA polymerase binds to the promoter, and the second step in the initiation is the first few phosphodiester bond form. So the number one phase, which is called initiation, has two steps, A and B. In A, RNA polymerase binds to the promoter, and the B is first few phosphodiester bond forms. And next step is called elongation. In this elongation happen, we can see that the more and more peptides are joined, and then termination, which end the transcription. As we will see now, the RNA polymerase in the initiation recognize a region which called promoter, which lies just upstream of the gene. So this is number one in the initiation. Polymerase bind tightly to the promoter, causing localized separation of the two DNA strand. So the binding of the DNA polymerase cause the RNA polymerase cause the two strands to be separated. Polymerase start building the RNA chain, adding ribonucleotides, and after several ribonucleotides are joined together, the enzyme leaves the promoter and the elongation begins. What happened in elongation is that RNA polymerase direct binding of more and more ribonucleotides in the from 5 to 3 direction, from 5' to 3 prime direction. Movement of the polymerase along the DNA template causes bubble, which is of separated DNA strand, to move also. As DNA transcription passes, the two DNA strand reform the double helix. Difference between transcription and between DNA replication. There are two fundamental differences between transcription and DNA replication. First is the RNA polymerase only makes one RNA strand during transcription. It copies only one DNA strand in a given gene. This makes transcription asymmetrical. Replication is semi-conservative. This is number two is DNA melting is limited and transient during transcription, but separation is permanent in replication. The last step The last stage is termination. Termination analogous to the initiating activity. Promoter, these are the region are there are regions are at the other end of the gene that can serve to terminate the transcription. So we have terminators, we call them terminators. So these terminators work with the RNA polymerase to loosen the association between RNA product and the DNA template. So from that we can understand that there is a promoter in the beginning of the gene and there are terminator in the end of the gene. So as a result, the RNA deassociate from the RNA polymerase and the DNA transcription stops. So we have now transcription landmarks. Landmark to know the main point in this process. RNA sequence are written from 5 to 3, 5 prime to 3 prime, left to right. Translation occurs from 5 to 3 with ribosomes reading the message from 5 prime to 3 prime. Genes are written so that transcription proceeds from left to right from 5 prime to 3 prime. The gene promoter areas lie just before the start areas said to be upstream of the transcription. Genes are therefore said to be lie downstream their promoter. In summary, transcription takes place in 3 stages: initiation, elongation, termination. Initiation involves binding RNA polymerase to the promoter, local melting, and forming the few phosphodiester bond. During elongation, the RNA polymerase links together ribonucleotides in the 5 prime to 3 prime direction to make the rest of RNA. In termination, the polymerase and the RNA product deassociate from the DNA template. So this is our summaries, and here's the practical on that lecture. I hope students start to answer this question. Describe the differences between. We have to know the difference between the transcription 3 stages, which is initiation, elongation, and termination. And also we have to know the difference in the protein domains, between protein domains and protein motifs. And on this lecture also you can get some idea about the difference between primary, secondary, tertiary, and quaternary structure of protein. I hope this lecture, you like this lecture. Please follow us on YouTube and start to see more good lectures in different languages about molecular genetics and other important topics. Thank you for listening, and please like and subscribe on YouTube channel. Thank you.